Hello, AP Bio. We're going to pick up where we left off in class, looking at how patterns of inheritance that Mendel observed were not quite as straightforward and simple as he reported, and that it's actually much more complex. So we're going to look at, we talked quickly or a little bit about pleiotropy in class, where we have one gene contributing to multiple phenotypes. And then we looked at how one gene can control another. Now we're going to look at how there are multiple genes that control a single trait. We call that polygenics, for more than one gene inheritance. So we have more than one gene, multiple genes affecting a particular phenotype. The classic examples of this are skin color and height. Now, the thing that you're going to look for with polygenic inheritance, if you look into a population of any organism, you're looking for a trait, or a character, I should say, that has a continuum of traits, meaning that there's this broad, or not necessarily broad, but there's a spectrum of possibilities. So let's talk first about skin color. Let's go in your notes and let's just clarify what polygenic means. So it's more than one gene pattern of inheritance, multiple gene affecting a single phenotype. And when we look into that population, we look at this bell-shaped curve. So just to clarify, this is a picture of phenotype. It's not a picture of genotype. So this is what the physical trait, how it might look in a particular population. And we call this bell-shaped curve a normal distribution. So you have most of the population here, and then you have some here and some here, but these would be the tails. And like I said, the classic examples are skin color and height. So let's look at what skin color might look like. Here's a normal distribution of skin color. When you look at a group of people, you'll see, or if we're talking about skin pigmentation in people, you see not 3 distinct color choices for skin, not 5, not even 15, but this continuum. There's no broken areas, all kinds of shades. And that's because skin pigmentation is controlled on many different genes. We see the same thing in height. We don't see people that are 5 foot tall and people that are 6 foot tall. We see people that are everywhere from 5 foot tall to 6 foot tall and everywhere in between. Now, this is the diagram of a study that was done, completed, I think, around 2008. And at the time, this is showing the color regions show areas that are of the genome, the human genome, that are implicated in having an effect on stature. So stature is what we call height. Now, like I said, in 2008, we were thinking that there was probably at least 44 genes that were associated with stature. In a study published in 2014, we now know that there are at least 400 genes associated with stature. Woo! So you can see why there's not people that are just 4 foot tall and 5 foot tall. We have everything in between. So this is showing you an oversimplified and outdated study where we have more than 400 genes associated with height. Now, we often get something called the additive effect, because each gene adds some type of a unit. So if you could think about each of those genes having multiple alleles, and let's say that there are short, medium, and tall alleles. So every time you get that tall allele, you get taller and taller and taller. And you can think of it's called the additive effect. And if you look at, this is a general diagram of what the additive effect looks like, but the dominant allele in this particular figure is showing you that as the capital letters are going to push you to the right of whatever this diagram is showing you. So say this was height, and you'd get taller and taller and taller the more tall alleles you had. But consider there's 400 of them, so taller and taller and taller. The chances that you'd have all of the uppercase, the tall gene, the tall allele, would be relatively unlikely. Most people would have an interesting mix of in between if you looked at the math. And so that's why you end up with this normal distribution. Same thing happens with skin color, and you can see it in this diagram right here. It's the additive effect. So you'd have light, medium, and dark allele options for every position for skin pigmentation, every locus that's associated with making your skin a particular color. Okay, so that's polygenic inheritance. Now, we have more than one gene affecting a single phenotype. Now, we also have situations where it's not just genes that are affecting phenotype. We have genetic factors, so multiple genes sometimes only single or just a few, but sometimes multiple, so a polygenic trait. And then also coming into play environmental factors, and we call these particular phenotypes that are associated with both genetic factors and environmental factors, we call them multifactorial because they have multiple factors that contribute to whether or not a particular phenotype will be expressed. A common example of a multifactorial disease is coronary artery disease. So that's why when you go to the doctor, they ask you about your family history, because likely buried somewhere in your family history is some kind of a faulty gene. So we have this phenotype as a combination of both these genetic and environmental factors. And the genetic result is you get what we call a predisposition. Did your grandma have it? Did your uncle have it? Did your grandpa have it? Maybe you have buried in there in your genome some type of a faulty gene or combination of faulty genes. But like I said, you might not present with the disorder unless you lead a particular lifestyle. We know some about multifactorial disorders from studying twins that have grown up in different environments. Twins that are separated or just adult twins that don't have the same lifestyle choices. They definitely have shared genes, but they have different environmental factors that are going to impact whether or not they present with a particular phenotype. Now, I keep talking about disease. We talk about multifactorial disorders a lot. A lot of times this language is associated with disorders, but it doesn't necessarily have to be something that's wrong. It could be whatever phenotype that's affected by both, like I said, both some type of underlying genetic predisposition that's likely affected by multiple genes, and then different environments are going to cause different phenotypes to be expressed. We also have interesting environmental effects on whether or not a particular trait is going to be an advantage or not. And we're going to talk about sickle cell anemia when we talk about evolution. The beta globin gene is coded for on chromosome number 11 in the human genome. And you can have normal beta globin, or you can, which is shown in the orange right here, or you can have a mutated beta globin. And when you have a mutated beta globin, you get funny shaped red blood cells because of the way the proteins crystallize together, and in low oxygen environments, the red blood cell kind of sucks in to one side. And in some environments, this mutation would be selected for. It would be an advantage. Okay, but that's another conversation. I just want to mention it now because we're talking about multifactorial disorders. Okay, now, or I should say, I should clarify, this is an environmental effect that's going to affect the frequency of that particular recessive allele. So it's not a multifactorial disorder. We'll talk about it later. Just hold the conversation for now on sickle cell anemia. Okay, now we, again, thinking about the complexity and sort of moving on past Mendel to the next guy, this guy, Thomas Hunt Morgan, and he did these exhaustive studies with fruit flies. So Mendel studied garden peas, and Thomas Hunt Morgan was 1907. So he was a little bit after. We were 1850s to 1907. So they were both alive at the same time. But Thomas Hunt Morgan was after. And we know from our conversation the other day that Mendel came up with the particulate hypothesis. But he didn't know that those particles, now we know they're gene loci, were connected to each other and carried multiple thousands of them on the same chromosomes. He didn't know that. And it was the observations of Thomas Hunt Morgan that Thomas Hunt Morgan came up with what we now know is the modern particulate theory of inheritance, the chromosomal theory of inheritance. he did, like I said, his studies looking at Drosophila melanogaster. So he used the fruit fly, and this is not the fly, the little buggies that buzz around your fruit in your fruit bowl. These are from, these are different. Those are actually vinegar flies, but we'll talk about that later. But we call these guys Drosophila for short. And the reason he used them is because, first of all, we like to use insects because nobody cares if they live or they die, which is not very nice, but anyway. And then he also used them because they only have 8 chromosomes. So their twin number is 8. And the chromosomes are really big, so you can see them underneath the microscope. And then also, he was able to get them to mutate really easily in the lab. And he could mutate them so that they had the particular traits were either the wild type, which is the type that's most commonly seen in the wild, that's typically dominant, or the mutated type, which is he would make it do something else. So you could follow particular phenotypes as you did the pairings and look at them through the different generations, through the parent generation and then the F1, the F2, and so on and so forth. And what he noticed was, what he observed was, in one of his studies, he saw that there were males that had both red and white eyes, but there was very few females, almost no females, that had the white eyes, many, many more males. And he began to think, I wonder if being female is somehow associated with having the inability to have the white eyes, and being male was somehow associated with being able to have the white eyes more often. And that's true. And now we know that as sex-linked. He came up with this idea of sex-linked traits. And what sex-linked traits means, all that it means is, well, first of all, he was tracking and observing biological sex because it's a really obvious phenotype. These are females and these are males. But all that sex-linked means is that there's a gene located on a sex chromosome. Now, we usually say that the gene is located on the X chromosome. Here's a micrograph, an electron micrograph of the sex chromosomes in humans. And we can see that the X chromosome is much larger than the Y. These are metaphase chromosomes, so they've been replicated. Here's the centromere, here's the Q arm and the P arm. And you can see the waist where the centromere is. But the X chromosome is much larger. You can see how small the Y is. And so when we use our symbol conventions, we show that the trait, the character, is carried on the X. So we use an X, a capital X, and then we show the symbol in the superscript. So if we were looking at a recessive disorder, now not all X-linked traits are recessive, but that's what I'm going to talk about right now. So a recessive disorder would mean that the normal would be capital N, the normal phenotype, and the affective phenotype would be lowercase n, just like we used that naming or symbol convention before. And if we looked at what that might look like, a classic example of that is hemophilia. Oh, I haven't shown you that there. And we'll do some practice problems on Friday in class. I just want to get to talk about what sex-linked means. And and here is what hemophilia looks like in a population. So we call it, like I said, it's an X-linked recessive disorder. So the males for an X-linked recessive disorder are hemizygous. In order to present with a disorder, all they need is the recessive allele. They don't have to, they have no, if they have the recessive allele, they're going to have the disorder because they have nothing to mask it. Now, if we look at the pairing, though, a mating of an X-linked recessive disorder, we see that an affected father will never have affected sons because he always gives the Y to his sons, and the disorder is not carried on the Y, it's only carried on the X. So if we look here at what the pattern of inheritance is, an unaffected mother and an affected father will always carry 100% of the time, they'll have unaffected sons, and always affected daughters, but not affected by the disorder. They'll just be carriers. Now, if we looked at a carrier mother and an unaffected father, then you could have an affected son, because 50% of the time she's going to give her her recessive allele to the son, and 50% of the time she's going to give her dominant allele to son. Okay, that's all for now. We'll look at sex-linked. We'll start with sex-linked on Friday in class. I hope this video was helpful.